How do you think life started? I don't really know either, but someone who does know is Nick Lane, who's a scientist who's been researching this for decades. And I interviewed Nick about this, and I want to give you his special behind-the-scenes take on how life began. It's a story both about information and about energy. Let's dive in. Hey guys, welcome to the channel. My name is Reese. I used to teach tech ethics at MIT, but now I'm teaching the series on how everything evolved, which synthesizes a decade of my research, including these 20,000 note cards I've made on all these subjects. So today we're talking about the origin of life, and specifically, we're going to talk with an expert about it. There's this amazing dude, Nick Lane, who I interviewed a couple years ago for my podcast, and I've read a few of his books, and I want to share with you some of the learnings that I got from our conversation. And so you can see it here, there's two sides of the origin of life. One side is this information side, and the other side is this energy side. And I came into my conversation with Nick being kind of information pilled. I was like, oh man, I'm writing this book on what information wants, and we're gonna talk all about how origin of life, information matters so much. And he both reframed my understanding of information, where he, instead of RNA-based information, he talked about information before that, which is really important, thermodynamic information. And then he also reframed my mind to say it's not really about information, it's actually more about energy. So I want to share some clips with you that help us learn these things. Okay, so let's talk about the first one first. Information. I came into this being like, oh, it's an RNA world, right? There's got to be some kind of RNA world. Once you get that RNA world, you have a survival of the fittest, RNA, and then that eventually creates cells and creates everything for life. That's how I came into it. And what Nick showed me is that RNA world are these sequences that happen much, much later in the origin of life. I think about something like this. Oh, it's this string of nucleotides, and there's A, U, C, G, and there it goes. And these things aren't that crazy, and they can kind of get built up over time, and which sequence wins, which is kind of true. But actually, it's more complicated. In order to create RNA, an RNA molecule, even just a single nucleotide, a single letter of it's super complicated. This gives you some of that instinct where it says, oh my God, here are all these different base pairs that are coming off of it and why they twist the way they do. That's slightly helpful, but even this, the whole base pairs, but then the blue is the backbone, which is also super complicated. So you see this, and this is the backbone of it, where you can see that not only are these base pairs, adenine, guanine, whatever, beside that are these very complicated carbon backbone with a phosphate group. This is it's like 40 atoms in here. It's very complicated to create something like this, much less start to string them together. And so the RNA world is actually a sequence, and it's a very complicated long sequence. And so what Nick taught me is that there's other kinds of information. You can just have simple information in the structure of these atoms, in the structure of these molecules rather. And you can also have information as they start to interact with each other. And that these things can happen when it's only like 2 through 6 carbons versus something like an RNA. Even a single nucleotide has dozens of carbon atoms. A chain of them has tons and tons of carbon atoms. Okay, so let's check out this video with Nick. And you can see there's me, there's young Reese over there a few years ago. I'm just gonna put this over my face so you don't need to look at me. And let's listen to Nick talk about other kind of information, a simpler kind of information in structure and interaction networks before we even get to RNA.
It's much harder because there's more steps involved to make the building blocks of RNA or DNA than it is to make fatty acids or amino acids. And fatty acids, they make up the membranes. And we and others have shown that if you just mix fatty acids in water and kind of shake it up a bit, you get what I like to call proto-cells, which is to say you get a membrane around an internal enclosed aqueous space. And they're very dynamic. They can grow, they can divide, they can fuse together. They're constantly moving and doing things. They look quite lifelike, even though it's just a chemical system that you're looking at. And the amino acids, they do all kinds of amazing things as well. They interact with minerals and can convert them into things like iron sulfur clusters that are involved in fixing CO2 to make more organics, including more amino acids. So there's positive feedbacks. You mentioned autocatalytic loops. The positive feedback is an autocatalytic loop. And so if you take this seriously and you start thinking through, okay, what steps do we expect to happen first? What would come afterwards? Nucleotides are quite late in the game. And to get to them, you've really got to, it was quite possible to think about replicating protocells with a proto-metabolism, which is basically thermodynamically governed, that this stuff just happens spontaneously and is capable of making the nucleotides and is capable of getting better at making the nucleotides through positive feedbacks. So we end up with a kind of non-living system, which are replicating protocells that are quite lifelike in many ways, but has no genetic information. It has some information in the membrane and in the metabolism, which is a thermodynamically encoded information, you might say, which says this is, and that's deterministic. That says you can do this, but you can't do that. And you're never going to be able to do that because, thermodynamics says you can do this. If you've got a different catalyst, maybe you can do that, but where's that catalyst coming from?
So that's great. So Nick is saying, yeah, you have thermodynamic information, which is different than sequence-based information. And he also does a great thing where he talks about thinking in terms of protocells, proto-metabolism, proto-RNA. You have all these smaller building blocks, the monomers, that have all this feedbacks that they're incorporating there, that then there's information in the system, but it's not sequence-based information. We don't need sequence-based information, but we have information in the structures and in their interaction networks. And this is the kind of proto-information and thermodynamic information.
Now, I've got these growing protocells. I'm going to introduce. I've got nucleotides forming in them. What happens if I allow them to polymerize for whatever reason? Now I have a random string of RNA, random letters, no information content whatsoever. But we know in the code that there are patterns in the code that suggest there's non-random interactions with amino acids. Hydrophobicity, for example, size is another one, that if you've got a random sequence of RNA, you will have interactions with amino acids that bind to that RNA for physical chemistry reasons. And if they bind, they're more likely to polymerize. And so you're templating from a random RNA sequence, you're templating a non-random peptide. And that non-random peptide will have function in a growing protocell. It will either help its growth or hinder its growth. And so from this point of view, I've just introduced information into biology without doing anything other than introducing a random sequence of RNA. And so the question of the origin of information goes back to the thermodynamic questions of why the hell did I just, where did all this chemistry, this prebiotic chemistry, proto-metabolic chemistry, why is it like that? Can we see the answer in carbon chemistry? Is it somewhere else? It's hard to say, but it's that's where the information comes into biology, is in thermodynamics, I would say, which is why I have to be a physicist.
Yeah, exactly. I love that. Yeah, it is. You can't just be a chemist. You have to be a physicist because you need to understand thermodynamic information. And so what Nick is saying is that what we have later, which is the ribosome, which takes as input an RNA sequence and then directly turns it into amino acids, which then fold up into a protein. He's saying that even before all this, before we even had this, the codon chart, which determines which RNA sequence will then turn into which proteins, that a reminder that the ribosome doesn't exactly look like this simple thing here, this little black box. It actually is this very complicated thing composed of all these RNA and protein sequences. And that when RNA and proteins start to interact, they have all these kinds of interactions that they like to do with each other or don't like to do based off of hydrophobicity and polarizations, things like that. So one thing I learned from Nick is that when we think about information, we shouldn't always think about sequences, but we should think about structure and interaction networks at smaller scales and in terms of probability distributions of what are the gravity wells and the natural ways that chemistry and physics like to operate. And then everything else beyond that, sequences and things, get kind of built on top of that simpler background condition or that simpler substrate. Okay, now let's talk about energy. If we think about our definition of life here, is the NASA definition. Life is a self-sustaining chemical system capable of Darwinian evolution. That system needs to have information and energy and matter. And so before, I used to think about life as mostly being some kind of, DNA looking thing like this. But in fact, we should start to see life as energy. And energy is hard to see. It's hard to conceptualize. We can start to think about these little cells as these kind of energetic sacs that keep electric potential around them. They use energy of that electric potential to spin a motor. And so starting to think in terms of energy was really, really helpful for me. And the first thing I want to share is this great other conversation between Michael Levin and Nick Lane. And Michael Levin's another great scientist who's been on my podcast as well. And there's a conversation between them talking about what they probably agree is the core to life. And the thing that they agree on is resting potential. Let's check it out.
I think both Mike and I would probably emphasize electrochemistry, or at least is borderline physics, really, in terms of what we're talking about. And so the key, I suppose, in my mind, is not a single moment where you switch from one thing into another one. Even when you introduce information, there isn't a single moment. But there's a process, and that process is partly over time and it's partly structured. And the thing that I like most, I think, about the kind of environments I talk about for the origin of life, which are mostly hydrothermal systems. But the nice thing about those systems is really that they have a structure which is cell-like, and they can template cells that, in effect, can grow within them. And when I say cell-like, the key attribute in my own mind to cells is the charge on the membrane. It's really that defines the cell from its environment. And which coordinates everything that's happening inside the cell. And so at the origin of life, you have effectively a structure with charges on barriers that are cell-like structures. And it gives you this as a setup from the very beginning. And then there's lots of interesting questions that we haven't really answered yet. But how does charge organize things? It certainly does, but to what extent and in what ways?
That's great. And so what Nick is saying here is that we have a electrochemistry. They're both into electrochemistry, and when Nick thinks of a cell, it's not just a pure barrier of a cell, but it's also the electric potential that surrounds a cell. And so when we think about a cell, like this little prokaryotic cell, you can't really see this here, but the key part about this cell is that its whole membrane on the outside, part of it's protecting itself from things, but it's also keeping an electrical charge. And that's its whole way that it gets energy, is it pulls in things, it rips stuff, it makes an electrical charge and then uses that charge to create ATP that then charges other things. And so that charge is the fundamental thing at the start of life, is it's keeping its own little energy bank there. And it goes out into the world to get more energy, to keep itself charged, and then uses that charge for other things. Now let's hear Michael Levin also talk about this, and he does such a great job of it as well.
Which is what Nick already mentioned, which is resting potential. And the thing that one interesting thing about resting potential is that in many ways it's a coarse-grained sort of higher level entity, because it's different. If you are a mechanism that keys off of resting potential, it is not the same as keying off of potassium concentration or sodium concentration or chloride concentration, right? It's this slightly higher level thing. And so if you're in fact, it's one of these things, like many other things in cognitive science. If you want to be a micro-reductionist, you might say, well, there's no such thing. You might say, right, if you're sort of a Laplacian demon kind of creature, right, you might say there's no resting potential. What there is there's some sodiums and there's some potassiums, and those are your low level kind of bedrock. But what's cool about resting potential is one of many things is that it's one of the first steps up that I think life does in generalizing, coarse graining, getting good at noticing large scale control elements that are not reducible down to individual details, right? And while all of these, of course, the resting potential is of course a consequence of the various ions you have, the fact that you can have the same resting potential from different concentration of ions, right? As long as you change them out correctly, right? So you can vary the low level details and still get the same resting potential. And conversely, you can get different resting potentials with exactly the same genetics, for example, with the same ion channels, because they could be open or closed. So you get this interesting bow tie kind of architecture where there's this like functional node in the middle where things sort of feed into it, and then multiplicities come out of it. But it itself is this super interesting node from the perspective of information theory, control, and all of that.
And so what Michael is saying is that there's this resting potential, which is another term to just say that there's a potential difference. It's kind of like a dam, but electrical dam that these cells are keeping there. It happens with our neuron cells. It's how we think. But it also happens with mitochondria. It happens with prokaryotic cells at the beginning of life, how they kept a resting potential. They kept their little charged membrane Resting potential is just another word for a charged membrane. And initial creatures were able to both sit in an energy gradient and an existing potential, use that energy flow to then start to do all of this work. And then after they did all that work, then they said, okay, wait a second, I want to keep this energy flow and this resting potential for later. And so they started to wait around and start to create it themselves. And that's when we started to get these little charged cells all around the world. So let's listen to Nick Lane talk about the thing that's upstream of resting potential, which is the Krebs cycle is the thing that creates resting potential. So let's hear him talk about that.
What's actually going on there? Well, what's happening in our own mitochondria is we're taking food. Glucose is the usual easy example to give, but it can be fats, it can be amino acids. There's lots of it, and they get fed into the Krebs cycle. They get broken down into carboxylic acids. So the Krebs cycle is made up of these carboxylic acids, and you start out,
which is 2 to 6 carbon, 2, 3, 4, 5, 6 carbon chains.
Fusing a very simple, two fairly simple ones together to make a 6 carbon string. It's called citric acid. And sometimes it's called the citric acid cycle, or sometimes the tricarboxylic acid cycle. But anyway, you got this molecule, it's 6 carbons long, it's got lots of oxygens and hydrogens bound to it. And then effectively what the Krebs cycle does is, well, it yanks out a carbon dioxide. It yanks out a carbon, one of the carbons with 2 oxygens on it, that we breathe it out, it's waste. And then it yanks out a hydrogen or 2 hydrogens. And now those hydrogens are attached to other molecules, NAD, for example, nicotinamide. I always get my names muddled up, but adenine dinucleotide. But the point is, it's hydrogen. We're dealing with hydrogen atoms. And you can think of it as hydrogen gas almost, but it's never hydrogen gas. We're dealing with hydrogen atoms stuck to other things. And in respiration, we feed these hydrogen atoms to oxygen. That's basically what we're doing. We're burning hydrogen in oxygen. It's rocket fuel. We're powered by rocket fuel. So what happens? We're not releasing all that energy as heat. We're actually conserving it. And in effect, we're splitting the hydrogen atom into an electron and a proton. So a hydrogen atom that's basically it. It's 1 electron, 1 proton. The electron gets fed to oxygen. The proton goes across the membrane. The energy that's released powers the extrusion of a proton across the membrane. This sounds very technical, but basically what you've done is you've just put a positive charge over there and left a negative charge over here. And so you've just generated an electrical membrane potential. You've got a voltage across that membrane. So we've got an electrified membrane now, and that's what you're doing. So the Krebs cycle is spinning around, it's stripping out CO2, it's stripping out hydrogen, it's burning the hydrogen and oxygen and using the power to electrify the membrane. It's kind of as simple as that. Now, that's what we do. That's what all animals do. It's what a lot of bacteria do, but some bacteria do the opposite. And the older we go, so we're feeding the hydrogen to oxygen, but what happened before there was any oxygen? Well, think about, we've got a cycle that's going around, and it's stripping out CO2 and hydrogen to electrify a membrane. Now, in a hydrothermal vent, where you've got lots of hydrogen and CO2, well, and you have electrified barriers around protocells, then we actually have the opposite. We now have a reverse Krebs cycle. It's taking CO2 and hydrogen, it's binding them together to make these things. It's doing it repeatedly, and it's being powered by the electrical charge on the membrane. So it's a reverse Krebs cycle, and it turns gases into the fabric of life. It's fantastic.
Yeah, so that is super cool. So fundamentally what Nick is talking about is this, where you're able to push, down here, you're kind of pushing hydrogens below and then keeping the electrons above. So you have a charged membrane, and those hydrogens want to get back to those electrons, and so they go through the ATP synthase, which is spinning a rotor to shove a phosphorus onto ADP to make ATP. But the key thing is this electron membrane. That's what Nick is talking about, is that we have this charge potential. Today, we do something where we'll take, a 6 carbon chain, glucose, citric acid, whatever, we'll shoot off the CO2, breathe it out, and then use the H2, pull the H2 towards the oxygen, rip it down, turn the hydrogen is a proton and an electron. And so you rip them, you make this charged membrane here. So you're making energy from biomass. But he's saying that back in the day, what was happening is we did this the other way. Today we do it this way, but back in the day, we did it the other way, where we had all the CO2, all of this hydrogen around, we had some of these charged membranes, and we went backwards, and we took the gases of the ocean, and we kind of started to synthesize biomass and turn them into these carbon chains. So that is the Krebs cycle. And these are the beautiful things I learned from Nick Lane, that you can learn about that information can be thermodynamic information, not just sequence-based information like RNA, and that energy, when we think about life, we shouldn't just think about DNA. We should think about energy and resting potential and these charged membranes and how we existed on a charge in the past and used that energy to synthesize life. And that these days what we do is we take these big carbon chains and we use those to create a resting potential for life. So I hope this helps give you an inside view of Nick Lane's mindset on the origin of life. And he's actually releasing a new book on this. So I'll let you know when that comes out soon. And until then, thanks and bye. Thank you as always for watching. And thank you to Roots donors for supporting this channel. If you want to understand more about the past and how we got here, check out the previous videos here. And if you want to understand the future and where we're going, feel free to subscribe here. Hope to see you then. Bye.